Good morning student. I am DR.J.SUNDARI, Head and Assistant Professor of Botany, A.D.M. College for Women, Autonomous, Nagapattinam. Now I am going to talk about the structure, reproduction and the life cycle of Lycopodium. Systematic position. Pteridophyta, division Lycophyta, class Helicolapsida, order Lycopodiales, family Lycopodiaceae, genus Lycopodium. There are about 180 species in this genus. Next, distribution and habit. These species of Lycopodium are worldwide in distribution. They are mainly found in tropical and subtropical forests. They are very commonly found on heaths and on humus soils in moist sandy places. In India, they are found in hills of eastern Himalaya. Habit. Lycopodium found in woodland in moisture and acidic soils. Epiphytic. For example, Lycopodium phlegmaria, erect and shrubby. For example, Lycopodium clavatum. Creeping forms. For example, Lycopodium cernum. Gives out erect branches at interval. Erect branch, highly branched and bushy, found in open woodlands. This slide shows different types of Lycopodium species. Lycopodium clavatum, Lycopodium volatile and Lycopodium phlegmaria, Lycopodium serratum and the last one Lycopodium inartatum. Next, this slide shows Lycopodium cernum, dicosmally and branch grows equal or unequal. Lycopodium root. The Lycopodium root first formed is ephemeral. Older plants have advantageous roots. They arise from the pericycle or endotomies. Stem. Weak, slender and rhizomatous, erect or pendant, creeping in others. Branch basically dichotomous, sometimes monopodial. Stem or branch covered with leaves. Lycopodium leaves. Different types of leaves. This simple, sessile, skull-like and small with median vein microphyles. Spirally arranged and dense on stem. Decussate or hold arrangement. Lycopodium leaf. There are two types of leaves, isophilous and anisophilous. Isophilous leaves, all leaves are of same type. Anisophilous, different types of leaves. In Lycopodium volatile, 4 rows of leaves, 2 lateral rows of large and falcate leaves, 1 ventral rows of small hair-like leaves, 1 tarsal row of medium-sized acicular leaves. This slide shows anatomy of root. This is outer layer of epidermis, middle layer of cortex and inner layer of stele. The outer layer of epidermis, single layer, thin wall cells, sometimes with unicellular hairs. Cortex, just below the epidermis, composed of parenchymatous cells. Older roots have thick walled cells formed mechanical support. Stele, protostele, monarch, diarch and triarch. Xylem is curved and phloem lies in between the arms of xylem. Anatomy of stem. 3 zones, epidermis, cortex and homogeneous. Epidermis, productive layer, cells with thick outer wall with cuticle. Cortex, nature varies with special and stem diameter. Homogeneous, cortex is entirely parenchymatous and heterogeneous, 3 zones, outer chlorenchymatous, middle parenchymatous and inner sclerenchymatous. Endotomies, distinct young stem, stele, protostele. This slide shows different types of steles. Flectosteles, actinosteles, protosteles and mixed protosteles. Protosteles, primary and xylem and phloem. Xylem surrounded by a phloem. Actinosteles, Xylem star-shaped phloem between arms of xylem. Protosteles in the form of plate-like structures. And mixed protosteles, xylem and phloem uniformly distributed in transverse sections. For example, Lycopodium cernum. This way shows anatomy of leaf. Different types of leaves. The leaf triangular in outline, outer epidermis covered by a thick cuticle. Stomata are present on both surfaces, ambistomatous in Lycopodium complenatum and Lycopodium olophylle. The stomata are hypostomatous, confined to lower surface. Mesophyll is undifferentiated, made of chlorophyllous cells. A median centric vascular bundle with a xylem surrounded by a phloem. Reproduction, vegetative reproduction, gamma of bulbils, fragmentation, formation of resting buds, formation of root tubercles, formation of advantageous buds, bispore, homosporous conditions. Vegetative reproduction, gamma and bulbis. The lateral outgrowth from the stem takes place of leaves. It consists of a short reduced axis surrounded by a thick fleshy leaves with storage food materials. The falls on the ground and grows into a new plants. Fragmentation, death and decay of older parts of the stem leads to separation of younger branches with grow into a new plants. Formation of resting buds. The tips of apical buds of rhizome and branches stored food material and is surrounded by a branch of leaves. In winter, rest of the plants dies and the rest buds develop into a new plants. This slide shows formation of root tubercles, group of cells with stored food material and product by thick walled germinates into a new plants. Formation of advantageous buds. In the leaves, the epidermal cells proliferate near base of and grow into a buds. These buds are capable of germinating into new plants. This slide shows Lycopodium stabilis. In Eurostasia, every leaf on the plant is sporophyll. In Rhopholastagia, the leaves near the apices bear sporangia and are called sporophylls. Sometimes sporophylls are distinctly smaller than the foliage leaves. Aggregation of sporophylls is called stabilis. A stabilis has a central axis on which spirally arranged sporophyll are present. This slide shows different types of stabilis. Next, position of sporangium on sporophyll. Auxiliary in position, for example, Lycopodium zelago and Lycopodium phlegmarium. On the dorsal side of sporophyll, epiphyllus, for example, Lycopodium cernum and Lycopodium clavatum. Subfoliar, not on the sporophyll, but the little towards the sporophyll. This slide shows Lycopodium abscissarum. This is longitudinal section of sporangium. This one stabilis, and this is microsporophyll sporangia. Next, Lycopodium stabilis, sporophyll and sporangia. This is upper side of sporophyll. Next, structure of sporangia. Reniforms are kidney shaped, has a stalk and a capsule. Capsule is unilocular. Sporangial wall is 3-layered. Innermost layer is the tapetum. Inside sporogenous mother cells undergoes meiosis and produce tetra of spores. Sporangia are homosporous. Next, dehiscence of sporangium and the elongation of the internode of stabilis. Sporophylls spread out, exposing the sporangia. A line of cells are differentiated on the sporangial wall are called stomium. The inner wall of the stomium are thick and lichenified. The exposed sporangia lost water and dry split appears in the stomium and the sporangia open into two valves. Disseminated the spores. Germination of spores. The time taken for germination varies from a few days to several years after the revelation from the sporangium. The quickly germination spore produce aerial short, live and green prothali. For example, Lycopodium serratum and Lycopodium inundatum. When spores take longer time to germinate, they get buried under the soil and produce the colorless subterranean prothali that are large, tuberous and long-lived. Next, the spores germinate to produce the gametophyte. The gametophyte is called the prothallus. It produces the anthraeal and archegonium. It is free-living, independent the sporophyte. The two regions, lower region attached to the soil and upper region generative zone containing the anthraeum and archegonium. Gametophyte. There are three types of prothali present here. Lycopodium cernum first type and second type clavatum type and third one Lycopodium phlegmaria. This is Lycopodium cernum type. The Lycopodium cernum and Lycopodium inundatum, erect, cylindrical, body 2 to 3 millimeter long, grows on the surface of the ground, colorless, basal portion potted in the soil, lobed generative zone green and bearing sex organs at the face of the lobes, rhizoids restricted to lower ported portion. The endophytic fungus is present in the basal portion. The meristematic tissue present as a rim around upper part of the prothallus. It is independent and prepared its own food. This slide shows different type of prothallus. Different types of prothallus. First one is Lycopodium phlegmaria and the second one is Lycopodium clavatum and the second one is Lycopodium complenatum and the third one is Lycopodium clavatum. Next, Lycopodium clavatum type, spore germination is delayed for a long time. Thus the prothallus has a longer lifespan. Here the prothali are fleshy, non-green, totally sporophytic and completely subterranean and perennial in nature. Development takes place beneath the surface of the ground or within a layer of humus. The prothali are large and may be 2 cm in length. They may be top-shaped with a convolute margin of carrot-like structure. The top of the prothallus are lobed and the sex organs and the growing embryos are located on these lobes. All the gametophyte cells are parenchymatous cells. Next, phlegmaria type. The prothali are aerial but sporophytic in nature, grow on trees, trunks below the coating of humus. This type found in epiphytic species of Lycopodium. For example, Lycopodium phlegmaria. Here the spore germination is imitated and the gametophyte grow for only one season. The prothallus consists of a short tuberous central part from which a number of colorless, slender, cylindrical branches develop in irregular fashions. These branches bear sex organs and they are usually surrounded by a glandular hairs called paraphysis. Vegetative reproduction of prothallus by formation of gamma. The bone or branches of prothallus singly or in clusters. thick-walled or stored food material capable of parenations. By progressive death and decay of older parts, advantages buds arise from the injury parts of the lobes. Next, prothallus. Prothali are monoecious, male anthria and female archiconia are born on the same thallus or same Lycopodium plants. The anthridium usually sunken at the prothallus position visible by a small bulge has a single layered jacket on vapor column cells. As water enter the jacket, rupture and biflagellate anthrozoids are liberated. Prothali. The archiconium flag shape having basal ventral and a neck. That has 4 to 8 neck canal cells. The ventral has a ventral canal cells and on egg cells. Fertilization. This is when archiconium matured tip of neck cells split neck canal cells and ventral canal cells disintegrated and form of a mucilaginous substance. The anthrozoids enter the archiconium and fuses with the egg to form the zoospore. The diploid zoospore germinate and produce the sporophytic in nature. This slide shows a life cycle of Lycopodium. The uppermost layer produce sporophytic in condition, that is diploid in nature, and the lower portion produce gametophytic in nature, that is haploid condition. The Lycopodium produce cone 2 eggs, cone produce microsporophyll, microsporophyll produce microspore mother cell, microspore mother cell reduction division undergoes produce spore, spore produce prothallus, that is prothallus containing anthridium and archiconium. Anthridium produced sperm, archiconium produced egg, sperm and egg produce fuses with the zygote. Zygote gives rise to produce sporophytic generation, that is diploid condition. Thank you.